So now you may start, sir.
Okay.
Sets are ready.
They are ready.
The streaming will start in 1 minute.
Srikumar joining, eh? Joined. Dr. P.K. Srikumar ji. Japan, sir?
Yes, sir.
Dr. P.K. Srikumar.
Sir, Srikumar sir, no unmute you, sir.
1 minute, 1 minute.
Dr. P.K. Srikumar.
Yes.
Japan ji, Dr. P.K. Srikumar, please make him on. Okay.
Yeah, yes,
Yeah.
yes, madam.
Yes.
All sets are ready. You may start
Yeah.
now.
I think you can start, Sonu. 6:30.
Okay, sir. A pleasant evening to one and all. An efficient diagnosis is the first step of getting effective treatment. According to the World Health Organization, without medical devices, common medical procedures would not be possible. They are providing access to good quality, affordable, and appropriate health by enabling various health technologies to diagnose illness, to monitor the treatments, to assist disabled people, and to intervene and treat both acute and chronic illness. Medical devices is such a huge domain which is going to rule the future of pharma sector. In vitro diagnostics or IVD is one of the core elements of medical devices and it attains a large attention of the public with the arrival of COVID-19 pandemic. Rapid antigen tests, RT-PCR tests all became a part of our daily lives nowadays. In short, it was a lesson for us to realize the actual significance of various in vitro diagnostic tools. The World Health Organization defines the IVDs as the first line of defense against the possibility of the re-emergence of eradicated diseases. These are the tests that can detect diseases, conditions, infections, as well as to monitor a person's overall health to help cure, treat, or prevent the disease by testing a variety of samples that have been taken from the human body. When the IVDs are becoming such a significant domain as the frontline healthcare professionals, the pharmacists who are in close contact with the public health and the medical devices, we are supposed to be the IVD experts too. This recognition, the recognition of the need to educate our pharma community on IVD, Indian Pharmacy Graduates Association Kerala State Branch, Indian Pharmacy Graduates Association Central, and the Kerala Pharmacy Graduates Association are jointly organizing the scientific webinar on the topic Recent Trends in Quality Assurance in the In Vitro Diagnostic Reagents so as to make a strong awareness on IVD in the pharma community. The most respected pharma professionals, let me introduce myself. I am Sonu Benny, PhD Research Scholar, Amrita School of Pharmacy, Amrita Vishwavidyapeetham, Thrissur. I feel so blessed to be the master of ceremony of this glorious occasion, and I wish a hearty welcome to each of you on behalf of Indian Pharmacy Graduates Association Kerala Branch, Central, and the Kerala Pharmacy Graduates Association. Let's begin with seeking blessings from God Almighty. First of all, let's begin with prayer song. take a moment to remember the departed souls just before a day. Dr. A. Padmanabhan sir, one of the very first and a well-experienced pharmacy teacher of Kerala. Sir is a retired professor of pharmaceutics, College of Pharmaceutical Sciences, Trivandrum. Srimati Janagi, mother of our vibrant pharma regulator, Dr. Nishit MC. Shri Madhavan sir, the better half of our beloved teacher, Professor Kamalabai. On behalf of IPGA, KPGA, and the entire pharmacy community, let's take this opportunity to convey a deep sense of gratitude for all their efforts to our profession and to mark our heartfelt condolence. May God strengthen their families and may their souls rest in peace. I'm kindly requesting all the participants to take part in a silent prayer for a moment to think of these departed souls. Thank you. Let's have a glorious beginning and a warm welcoming. It is my privilege to invite one of our well-experienced and energetic pharma professional, Mr. Matthew Cockart, Managing Director, Cockart Pharmaceuticals, to welcome the gathering. With much respect, I invite you, sir, for the welcome address. Over to you, sir.
Hearty greetings to one and all present in this webinar on recent trends in quality assurance in the video and in vitro diagnostics reagents. This webinar, organized by Indian Pharmacy Graduates Association and Indian Pharmacy Graduates Association Kerala chapter in association with the Kerala Pharmacy Graduates Association, is an added further to the various professional activities conducted by them. Now, I am here to welcome the August gathering in this webinar. First, Dr. C. S. Sadish Kumar, chairman of this function, is the ex-trust controller of Kerala stage, known as one of the mighty department heads of the stage. He is also a well-known devotional and motivational writer. He is the founder president of IPGA Kerala and executive member of KPGA and many other professional associations. Now he is the senior vice president of Agape Diagnostics. I extend you a hearty welcome to you, sir. Next is our beloved Dr. Adil Kumar Nasa, Trust Controller and Licensing Authority, State of Delhi, is the dynamic president of Indian Pharmacy Graduates Association. He is a hardcore pharmacy professional, always using his vast contacts and multidimensional capabilities for the betterment of pharmacy profession. He has kindly consented to inaugurate the webinar. On behalf of IPGA Kerala chapter and KPGA, I extend you a very hearty welcome to you, sir.
Thank you.
Mr. PM Jain, thank you. Mr. PM Jain, Trust Controller Kerala Stage, has kindly consented to be the guest of honor of this function. He is one of the main pillars of pharmacy profession in Kerala, and with his profound leadership, pharmacy profession is gaining greater momentum in serving the people in this most catastrophic period of the pandemic corona. Sir, on behalf of all who present here, I extend you a hearty welcome.
Thank you.
Dr. P.K. Srikumar, ex-deputy Drugs Controller of the stage, a well-known pharma professional, a motivational speaker, and maybe the voice of pharma profession in Kerala, is the dynamic president of Kerala Pharmacy Graduates Association. He is here with us as one of the co-organizer and to deliver a felicitation speech. I extend a hearty welcome to you, sir.
Thank you, sir.
Mr. Sanjaymon, Associated Vice President of Agape. Agape Diagnostics is in the field of clinical diagnosis since 1998. He is a well-known technocrat and a scientist, and attended many international seminars and conducted more than 30 scientific sessions. He is one of the authority in medical diagnostics and diagnostic reagents and equipments. He is here with us to present the subject. I extend a hearty welcome to you, sir. Mr. Dilip Ji, moderator of this function, is the Joint Secretary of IPGA Kerala State Branch. He's one of the dynamic members of and organizer of professional bodies in Kerala. He is the General Secretary of Kerala Gazetted Officers Association, head of the State Drug Testing Laboratory, Tichur. Sir, I extend you a hearty welcome.
Thank you, sir.
Professor Jayapalan is the host of this webinar and our support in this virtual platform. Thank you very much for consenting to be here. And sir, we extend you our hearty welcome to you. Mr. P.U. Abdul Nasir, the most dynamic General Secretary of KPGA, is here with us to propose vote of thanks. He is a stalwart in pharma industry and a hardcore professional. We extend a hearty welcome to you, Mr. Nasir.
Thank you, sir.
On behalf of KPGA, on behalf of IPGA, IPGA Kerala chapter, and KPGA, I extend our warm and hearty welcome to all our dear participants in this webinar. Thank you, and over to Sonu. Thanks.
So much, sir. It is my privilege to invite our super active pharma regulator, Dr. C.S. Sadish Kumar sir, President, IPGA Kerala State, the former Drugs Controller of Kerala State, and Head Business Development Agape Diagnostic Limited, Cochin, to deliver the presidential address. With much happiness, over to you, sir.
IPGA National President Dunasaji, our Kerala State DC, JNPM, and Sri Kumar, and my dear colleagues and friends. I am much privileged to speak a few words on this occasion. The seminar on quality aspects of IVD reagents under the aegis of IPGA Kerala, IPGA Central, and KPGA. As you know, IVD segment is a very small part of the medical devices where majority of the clinical testings are made. All of you know that 85% of the modern diagnosis is based on the biochemical values or presence of biomarkers or DNA, RNA sequencing or molecular results from your blood, from urine, other body fluids or tissues. The IVD has become most prominent, and attracted the public attention with the emergence of our COVID pandemic, when most of us are compelled to test for COVID, and when our IVD industry also could scale up the production of diagnostic tools for screening and clinical testing for COVID within a short period when the demand was very huge. For example, when all the international supply chain was disrupted, and during the peak time of the COVID in India, when all imports were delayed due to the global lockdown, Agape Diagnostics Cochin has served our nation with more than 10 million unit tests of reagents of CRP, E-dimer, ferritin, the three most important COVID prognostic reagents to measure the severity of COVID-related pneumonia and lung infection. It was the 70% of our demand nationally. See, this is the beauty of the strength of our Make in India strategy when dependence on imports fails sometimes. So presently, the approved test centers of IVD reagents in India are very, very less. For example, National Institute of Biologicals, the NIB Noida we call, is authorized only for a few classes of reagents like blood groupings, dengue, malaria, chikungunya, HIV, HCV, SBSAG, etc., as well as ICMR approved center also. And again, new panels are coming added to NIB every day. Then come to virology institutes, of course, ICMR approved viral institutes are also centers. Then for oncology things, AIIMS and Delhi, AIIMS Delhi as well as Tata Memorial Hospital, Mumbai are also approved centers. Other chemistry parameters are approved by any NABL labs generally. So in future, once standards are laid down in the books of official books, all our state drug testing laboratories will be designated by government for testing the medical devices. And so today's the principles of QA/QC aspects today will be a prelude to our list of our laboratories in future. Today, we have very, very experienced faculty called Sanjaymon to speak on the quality aspects of IVD. I wish that all our drugs analysts, pharma, if opportunity to clarify their doubts today with Sanjaymon. We will come with the QA/QC principles of for equipment next time shortly. I think we are planning to have our talk on the equipments next time. So, sir, I wish you all, everybody, I wish all a good seminar. Thank you so much. Sonu, over to you.
So much, sir. It is our blessing to have Dr. Atul Kumar Nasa sir, the Honorable National President, IPGA, to inaugurate this function. Dr. Atul Nasa is working as Drugs Controller and Licensing Authority, New Delhi, and he is a member of various committees constituted by the government for framing, designing different policies. He has more than 25 years of experience in regulatory affairs. He is a recipient of Best Drugs Controller Officer Award and also the Fellowship of Indian Pharmaceutical Association for his distinguished and dedicated service in the profession of pharmacy. Dr. Atul Kumar Nasa has been honored with prestigious KC Chatterjee Memorial Award 2015 and also the IPGA Professional Excellency Award. He has also been entrusted the role of Vice President of All Indian Drugs Control Officers Confederation and Director of Indian Confederation of Healthcare Accreditation. With utmost respect and heartfelt happiness, I invite you, sir, for the inauguration and inaugural address. Over to you, sir.
Thank you very much, Dr. Sonu, for a nice introduction. And very good evening to all of you. I welcome on behalf of Indian Pharmacy Graduate Association, IPGA Kerala branch, and Kerala Pharmacy Graduate Association, all the participants to this webinar on recent trends in quality assurance in the in vitro diagnostic reagents. My thanks to Mr. Matthew Cockett, the Managing Director of Cockett Pharmaceutical, for the nice introduction. My also thanks to Dr. Satish CS, the President of IPGA Kerala branch. He is the ex-drug controller of Kerala state. My friend, the drug controller of Kerala, Mr. Jayan PM, Mr. Dilip Ji, the Joint Secretary of IPGA, Dr. P.K. Shri Kumar, Dr. Jayabalan, Mr. Abdul Nasir, and all other participants to this webinar. I welcome you all. Now see, during this pandemic time, when the entire world is suffering from COVID, we have seen the first wave, we have seen the second wave, now we are in the third wave. And during the second wave, which was typically, dangerous, and we have seen so many casualties. So this is the right time to know about in vitro diagnostics, what important role they play. Dr. Satish, in his introduction, has told that when the import was totally banned and we were not getting, medicines or APIs or other related, products from other countries. So it was our own homemade, companies, Atmanirbhar Bharat, self-dependence country, who played a very important role in pandemic time. And we know on one side, India is the pharmacy of the world. We export so many formulations to the entire from our country to the entire world. But apart from that, pharmaceutical industry or the formulations what we manufacture, the IVD industry or medical device industry is also very important. Government of India has brought in notification MDR 2017. So as on date, this MDR 2017 is applicable, and whatever the guidelines, whatever the rules, whatever the sections are given, the entire medical devices industry, IVD industry is working on the basis of that. Definitely in COVID-19 time or pandemic time, like rapid antigen test, RT-PCR test, they have played a very important role in detecting the infectious disease at an early stage. In immunochemistry, in clinical chemistry, in molecular diagnostics, in microbiology, in hematology, in coagulations, and particularly in hospitals, in clinical laboratories, in academic and research institutions, IVD are playing a very important role. So this is the right time to understand the quality assurance aspects of IVD reagents. And see, there are 2 points. One is quality control and other is quality assurance. Both are interrelated, but quality assurance is a way of preventing the mistakes and defects in the manufactured product and avoiding problems. So I think the today's webinar, the speaker of the day, Mr. Sanjaymon MR, who is the Associate Vice President, he will be discussing on various aspects on quality assurance in vitro diagnostics, which will be definitely helpful to all the participating members and particularly to the in vitro diagnostic industry. With these remarks, I inaugurate this webinar jointly organized by IPGA Kerala branch and Kerala Pharmacy Graduate Association, and also my heartfelt condolences to all the departed souls. So thank you very much, and my best wishes to all the participants for this webinar. Thank you very much.
Thank you, sir. Thank you.
So much, sir. It is our fortune to have the head of Kerala Pharma Community, the respected Drugs Controller and Licensing Authority, Government of Kerala, Shri PM Jain sir, as the guest of honor of the day. Shri PM Jain has started his career with Drugs Control Department in 1993 and worked at various capacities and was celebrated as an efficient pharma regulatory officer. Moreover, he is the one who is regulating the medical devices under which the IVDs are belonged to. He is the former General Secretary of IPGA Kerala State Branch. With much respect, let me invite Jain sir to deliver a few words as our guest of honor of the day. Over to you, sir.
Is it audible?
Yes, sir.
Thank you, Sonu. Good evening to one and all. Dr. C.S. Sadish Kumar sir, President, Indian Pharmacy Graduates Association Kerala branch, who is chairing this function. Shri Matthew Kokat, MD Kokat Pharmaceuticals. Dr. Adul Kumar Nasa sir, National President of Indian Pharmacy Graduates Association and Drugs Controller and Licensing Authority, Delhi, who inaugurated this function. Shri Sanjaymon KR, Associate Vice President of Agape India Limited, and key speaker of this evening, Shri Dilip G, Joint Secretary, Indian Pharmacy Graduates Association Kerala branch and moderator of the technical session, Dr. Jay Balan, Dr. P.K. Sri Kumar, Shri Abdul Nasir, General Secretary, KP Kerala Pharmacy Graduates Association, and other dignitaries present in this webinar. Fellow members of Indian Pharmacy Graduates Association, Kerala Pharmacy Graduates Association, and my dear friends. It is honored to be a part of this auspicious occasion, jointly hosted by Indian Pharmacy Graduates Association and Kerala Pharmacy Graduates Association. Professional bodies like Indian Pharmacy Graduates Association and Kerala Pharmacy Graduates Association has pooled their resources and talented to roll out this wonderful online training session for recent trends in quality assurance in vitro diagnostic reagents. The global in vitro diagnostic market size is projected to reach $96 billion by 2025 from $84 billion in 2020. The rising demand for point-of-care IVD devices has positively impacted the growth of global IVD market. The last 2 years was a setback for everyone due to pandemic COVID-19. But the COVID-19 pandemic had accelerated the decentralization trends in testing and how the POC testing is used as a key tool to manage healthcare capacity. The recently launched IVD instruments and devices are convenient to use and highly efficient, which has increased their adoption among healthcare professionals. Several OTC diagnostic products are available in the market now. These products are mainly preferred by elderly to reduce hospital visits. The importance of these products has increased during COVID-19 pandemic, as people are opting for convenient diagnostic tests that can be performed in home care settings. These factors, coupled with increasing prevalence of chronic diseases like diabetes, cancer, and contagious diseases like dengue fever, chikungunya, and increasing awareness about disease diagnostics and growing disposable income levels are expected to support the IVD market. The increasing geriatric population in developed as well as developing countries will positively influence the growth of IVD market. With rapid growth in geriatric population globally, it is estimated that 60% of individuals aged 65 or above will be living with more than one chronic condition by 2030. Pneumonia is one of the fatal infectious diseases that affect the elderly population in the age group of 60. Diabetes is another chronic disease that affects the geriatric population, since the population is prone to endocrine disorders due to hormonal imbalances. According to International Journal of Health Policy and Management, China is one of the countries with the highest prevalence of diabetes. Around 114 million people in China suffer from diabetes. According to the National Diabetes and Diabetic Retinopathy Survey, there are about 12% prevalent cases of diabetes in India, as diabetes is a major risk factor for many of the diseases. When coming to our topic for discussion, the maintenance of quality management system is a crucial for an IVD manufacturer for providing the correct results every time. The quality management system must include parameters like documentation, standard operating procedures, quality control samples, and external quality assessment scheme. The fifth schedule of Medical Devices Rule 2017 had laid down the standards for quality management system for medical devices and in vitro diagnostics in our country. I once again extend my sincere thanks to all organizers of this function for inviting me to this webinar. Thank you. Over to Sonu.
Thank you so much, sir. It is my privilege to welcome our multi-talented pharma professional, Dr. P.K. Srikumar, President, Kerala Pharmacy Graduates Association, and the former Deputy Drugs Controller, Kerala State Government, to felicitate today's event. With much happiness, over to you, sir.
Am I audible?
Yes, sir.
Yeah.
Okay. Good evening to all. Thank you, Sonu. Respected Dr. Satish Kumar, who is chairing the function, President of IPGA Kerala. Dr. Adil Kumar Nasa, sir, DC and Licensing Authority, Delhi, and National President of IPGA. Then today's guest of honor, Sri Jayan, Drug Controller, Kerala. Mrs. Sanjaymon, Associate Vice President Quality Control, Agape. Then Moderator and Joint Secretary of IPGA, Mr. Dilip. Speaker of today's session, Professor Jayapalan. General Secretary, Sri Abdul Nasir, KPGA General Secretary. Sonu Beni, stalwarts of pharmacy, dear friends and students. It's my great privilege and honor to be a part of this great event. In the banner and as the president of KPGA, this particular event aims to limelight recent trends in quality assurance in the in vitro diagnostic reagents. We know many of the medical devices and all in vitro reagents used for the diagnosis of disease and disorders in human beings and animals are drugs, and our medical device rule 2017 has become a landmark in the history of our Drugs and Cosmetics Act and rules, where our government also formulated a separate rules for the regulation of it, and the quality management system is in place of our GMP corresponding to our medicines. We know now devices play a very important and prime role in the diagnostic part, and without proper diagnostics, a doctor or a drug even can't act much. So our medical device market also stood at US dollars of around 10.36 billion in the financial year 2020, and expected to be around 50 US dollars by 2025. We came across a lot of devices, reagents, and kits, especially nowadays, which is very common because of the COVID pandemic. And of course, these should be reliable, dependable, accurate, easy to operate, and stable. And here lies the importance of today's topic. We, the pharmacists, may not be well versed. All, of course, we know thorough knowledge on our medicines, but devices part and I mean, including the reagents, we are not much having that much of knowledge. And of course, recently a similar webinar was organized by the associations of in Kerala, which was exclusively for the the devices and reagents. And today's speaker has handled a session on that. And of course, he is a very good speaker, and his knowledge is remarkable. His capability on presenting the subject is remarkable. Also a very senior officer who played a vital role in the accreditation of DTL Thiruvananthapuram, our Dilip, who is moderating the session. And thank you once again, the organizers, IPGA, Kerala branch, IPGA National Forum, and KPGA. And I'm very thankful for me to be a part of this wonderful program. Deep condolences to the pharmacy of the departed soul. Once again, thank you, organizers. Wishing the entire program a great success. Thank you.
Thank you so much, Srikumar sir. Here we start a brainstorming session, a hunt to treasure the multiple facets of the in vitro diagnostics. For the kind attention of all the participants, keep your questions posted on the chat box and feel free to have a discussion after listening to the aspects of IVD reagents. The link for availing e-certificate will be shared with you all at the end of the session. So directly we are entering to the most awaited moments. Let's begin our scientific session on recent trends in quality assurance in the in vitro diagnostic reagents. I feel so glad to see that we are going to hear from Mr. Sanjaymon KR, Associate Vice President Quality, Agape, the right resource for the topic. He's having an experience of more than 20 years in the domain of medical devices and the apt one for handling the topic. At this juncture, let me invite our moderator, an efficient pharma professional, Mr. Dilip Ji, Secretary, IPGA Kerala State Branch, and Analyst, the Office Head, Drug Testing Laboratory, Medical College, Thrissur, to introduce our eminent speaker of the day, as well as to moderate the scientific session. With much respect, over to you, sir.
Thank you, Sonu. Good evening, one and all dignitaries who is now present in the webinar. In the recent trends in the quality assurance in vitro diagnostic reagents, Sadish sir, President IPGA, and National President NASA sir, and all other eminent personality, and the speaker of the day, Dr. Sanjaymon KR. And because today this subject is more relevant, because during the COVID pandemic has struck all world. The in vitro diagnostic reagent role is more crucial. So quality control, how much so far is the quality control of in vitro diagnostic is done. So as a analytical session head, because I am not aware of much about this COVID pandemic, this in vitro diagnostic reagents. So all our analysts are here from all the three labs, all the chief government analysts from Trivandrum and the Ernakulam, and from my lab also attending the meeting. And many of the students from all colleges are attending the function because it is a new subject. So it is a great honor for me to welcome the speaker of the day on behalf of IPGA and the IPGA Kerala branch, and in association with the Kerala State Pharmacy Graduate Association, the Sanjaymon KR, State Vice President, Quality Agape Diagnostic. So I will have a small introduction on his side. After graduation and DMLT, he joined in Medilab Diagnostic in the year 1998 as a trainee technician extensively worked in the Department of Clinical Chemistry, Clinical Pathology, Hematology, and Phlebotomy and Serology section. From the year 2002, he was deeply involved in the process of getting the coveted enabled accreditation for the laboratory as a quality control coordinator. Started the career in Agape Diagnostic in the year 2006 as a customer support executive in Chennai. Attended major international conferences like Medicare Germany, ASC US, CME China, CACLP China, Medilab Dubai, Arab Health Dubai, author of GLP session in Tech Agape, the technical magazine of Agape from the beginning. Conducted more than 30 scientific sessions, including the session in JSS Medical College, Mysore on physiologic mixing, and in Nepal and Myanmar on quality attributed. We have training. He have completed training in ISO 17025, ISO 15189, TBA 120 FR, application training, Toshiba Medical System Corporation, Japan, ISO 15189 2007, Quality Council of India, CA 270, Clinical Chemistry Analyzer, Toronto Electric Company, Japan, EMS 301, Clinical Chemistry Analyzer, Tokyo Bouquet Medicines, including International, Tokyo, Japan. So we have a very vast knowledge regarding this quality control of the recent in the in vitro diagnostic and reagents and in the enable and all the international accreditation body. So with much pleasure, I invite him on behalf of IPGA, IPGA Kerala and KPGA have a nice webinar, because all persons are really waiting for the quality control aspect of the in vitro diagnostic, because it is a new subject for all of us. Thanking you. Over to you, sir.
Thank you, Pillai sir, for the wonderful introduction about me. And it is a pleasure to address such a vibrant gathering of I would like to say the stalwarts in the pharma industry. And so this session is basically into the IVD reagents. When I think from the inaugural note itself, the importance of IVD. So now to look at the IVD segment is growing at a faster rate, and because of the recent pandemic. So once the industry grows at a faster rate, we would like to say it's a revolution. Then the quality plays an important role, or else then it can go down at a faster pace. So today the topic is how we are assuring the quality in vitro diagnostics reagents. So I'm just so one is the common quality attributes. Then there is a small time for the questions or queries. Then after that we can go to the next session. So rather than going continuous, so we are having the sessions in two parts with the query in between. So just I'm going to start my to share. Sharing, please. Hello.
Yeah, you can share, sir. No problem.
Just a minute.
You are a co-host.
Yeah. Just a minute. Share screen. Yeah. Some problem. I think. There is some problem in sharing. Just a minute.
You can keep your video off, so that will be better.
Yeah. I'll keep my video off. Then I will again share. Say something. I think there is some problem with the sharing, I believe.
Share myself, Sanjay.
Just just one this thing, and then let me check once again. If it is sharing, then it is okay, or else you can share it from your side.
Sir, please check your network also. It may be slow so that it is less time.
Okay.
It takes time for transmission.
Okay. Let me once
We are
again.
screen.
Let me
We are seeing the screen, but we are seeing your blank screen only.
Just a minute. Again, I'm sharing now.
Okay.
Now, is it?
Yeah. Okay now.
Okay now.
Please, please make it as a screen, full screen mode or slideshow.
I'm keeping it in full share mode only, but I don't know why.
We
Actually,
are seeing
sir,
left side. We are seeing the other slides.
Actually, this is in the presenter mode. You make it as a full screen mode.
Just a minute. Once again, I'll share it because.
Yeah.
Let me. Sometimes. Now is it?
Yeah.
Yes.
Yeah. Okay.
Yeah.
Okay, sir. Done.
Okay.
Okay. Thank you. So today's topic is how we are assuring the quality in IVD reagents. So we should know what is an in vitro diagnostic test. In vitro diagnostic tests are the tests which is used to detect disease, conditions, or infections. And in vitro simply means in glass, which means that these tests are typically conducted outside the body, as opposed to in vivo, which is inside the body. The importance of in vitro test is that they allow doctors to diagnose patients effectively and work to provide appropriate treatments. So this is actually the major challenge. The IVD test or the results of the IVD test is so important today. If you look at today's scenario, more than 85 to 90% of the decision taken by any doctors today is depending on the in vitro diagnostic results. So that is the current scenario. Example is, the COVID. Today, if a person is suffering from fever, whether it is a common fever or a COVID fever, there are only two ways. Do a COVID testing and see whether he is affected or not. So the complete reliability on the IVD testing today. Forget about the COVID. If a person is suffering from, say, diabetes, again, the dependency is huge. If a person is suffering from some liver disease, even the dependency is huge. If a patient came to a hospital or an ER where he is having symptoms of a cardiac arrest, to know whether it is a cardiac arrest or simply something else, the doctor depends upon, something called as a cardiac triple marker. It is nothing but, high sensitivity troponin, cardiac, BNP, NT-proBNP, and myoglobin and CKMB. We call it as a cardiac triple. So if the cardiac triple is positive, it says, yes, the person is suffering from a cardiac issue, then immediately, based on these results, the doctor need to administer the medicine. So that is the importance of, the IVD today. When we look at IVD, IVD is having two, segments. One is a hardware segment, another is a consumable segment. The hardware means the instruments which is used to do the testing. It starts from a simple glucometer, then to instrument with 2,000 tests per hour throughput, in modular systems and different instruments used in different I would like to say departments in the IVD. IVD is having multiple departments. One of the department is a clinical chemistry department. The second is the hematology department. Third is clinical pathology department. Another is imaging department. When we look at the other segment, it is microbiology. Today, microbiology is very big. And then goes to histopathology department and molecular diagnostic department. And sometimes it comes also into nuclear medicine. So each department is having its own paraphernalia. For example, a clinical chemistry department, they're having either semi-automated or fully automated, I would like to say, automated systems based on the workload or based on the requirement of the client. When we look at the hematology or cell counters, again, based on the workload, the client may be having a simple systems to a complex system. Similarly, histopathology, there may be fully manual systems and there may be fully automated systems where the number of samples is more than they need to do fully automation. So whether it is automation or semi-automation or manual methods, whatever be the methods used used for the testing, assuring quality is the #1 aspect, or assuring quality of the test or validating the test. In the common term, it is called as validated test result is very important when IVD testing is concerned. And here there are two major factors are playing for the quality assurance. One is the hardware, which is the instrument, and another is the consumable, which is the reagents. Now, when we look at the reagents, the reagents may be strips or a glucometer, the strips which is used in the glucometer. It may be or I would like to say an RDT or rapid diagnostic test. It may be a cartridge, or it may be a wet chemistry or the chemicals which is used for automated instruments. When these two together actually is responsible for the quality, or actually is responsible for assuring the quality of test or validating the test. When we look at quality, here today we are talking about quality in a different aspects. I think most of us know that what is the important meaning of quality. It is degree to which a set of inherent characteristics fulfill requirements. So here the word inherent characteristics is of importance, which means that if I say that, okay, my reagent is having a quality of 1, 2, 3, or my instrument is having a quality 1, 2, 3, it is based on certain characteristics, or I would like to say quality attributes. So based on these quality attributes or quality characteristics, I'm telling that my instrument is having a quality standards based on this, or my reagent or consumable is having a quality standard based on this. Why I said like the characteristics is that when it comes to the hardware, it is an electrical equipment. So the quality standards is something like an IEC standard, or it is having a different standards itself. It is having a different quality system itself. When it comes to the reagent, it may be having a different standards itself, or a different system of testing itself. So that is why I said the inherent characteristics for hardware and the inherent characteristics for the consumables or the reagents is little bit different. But please understand, without a hardware, we cannot validate a reagent, and without a reagent or consumable, we cannot have validate an instrument. So it is the both the sides of a coin. It is a symbiotic relationship, I would like to say. Now, when we look at quality, what are we looking for? In an IVD test, for example, if you are going to a hospital or if you're going to a laboratory, what are you actually looking at? So, for example, I went to a laboratory, I got my sugar tested, I got my value, say 1, 2, 3. So, what am I looking at? I have 3 things in my mind. 1, whether this is accurate. 2, whether this is precise, and whether this is traceable. So, first is accuracy. I always ask, even a common man, when the common man go to any of the laboratory, while getting a report, he usually think whether this is an accurate report or not. So in an IVD test, accuracy plays an important role. Whether in inaccurate test sometimes can give big trouble to the physicians. Now we will come up with some examples. Another is precision. Whether the report is precise or not, because accuracy is something different and precision is something different. Then the third one is a resolution. Whether, see, resolution doesn't come when it is a gravimetric measurement. Resolution comes when usually when it is very important for, say, for example, imaging system. Now let us consider what is accuracy. Say accuracy means if my glucose level is 100 and 100, for example, I got a glucose level of 100, whether this 100 is correct or not, that is a question when we think about accuracy. So if you look at on the right hand side of the presentation, you can see 4 circles. One is accuracy, another is precision, and the third is precise but not accurate, and not accurate and not precise. These are the 4 major things which happens when anybody do any analysis, whether it is a biochemistry analysis or whether it is some other analysis. Accuracy and precision is of utmost importance. Second thing is a resolution. Say resolution means I'll just give you an example. These are 2 measuring scales are shown here. The first one actually it measures in kg, kilograms. This is used for measuring weight, like weight of a person, weight of some our normal, what do you say, pulses or grams of groceries. We can use this, no issues. Whether it is 0.5 kg, 0.2 gram, no issues. It is not actually matters. But we cannot use the same weighing scale for weighing gold. For weighing gold, if you look at the middle, we are using a different scale. Why we are using a different scale? Because here, in while weighing a gold, we require more resolution. For example, we require maybe in milligrams, or maybe we may be requiring picograms or something like that. So that is why here yes, resolution is important. It is based on the application. Which application are you using? Another one simple example is that, based on the resolution, the hardware also changes, and based on the resolution required, the consumable also changes. I'll give you a typical example of glucose and thyroid. Now we all know about thyroid and all. So for doing thyroid, the measurement unit is in nanogram or picogram per ml. Whereas when we measure a glucose, the glucose is measured in milligram per deciliter or gram per liter. That is a measuring unit. So the precision or the resolution required for the hardware or the resolution required for the reagent for measuring a glucose is much less than the resolution required for the hardware or the resolution required for the consumable for a thyroid function test. That is the reason these 2 instruments are using 2 different methodologies. Sometimes the methodology also change when we are going for a higher resolutions. In glucose, we measure in using photometry, whereas the thyroid or a hormone we use to measure using CLIA or chemiluminescence immunoassay, which is more I would like to say more sensitive to the assay. So any value when a laboratory gives, 3 things are there. One is accuracy, another is precision, and the third is resolution. These 3 things needs to be matched or at par so that the laboratory can say that yes, my result quality is assured. When a laboratory says my result quality is assured, then this quality assurance, a part of the quality assurance, I won't say the 100% of the quality assurance, a part of the quality assurance actually goes to the manufacturer who manufacture this reagent or who manufacture this instrument for detecting that particular analyte in the body fluid. Now, when we say validated, if I'm telling that as a quality head, I say that, okay, my reagents are validated. So what I validate in my reagent? I need to validate something. Na? I need to validate. I'm saying that, okay, my reagent quality is assured. I put a quality QA seal. I put a seal that QC passed. So when I say that, my reagent is QC passed, how I validated? What are I validated? And what are the attributes to be validated? For reagents, there are some attributes like physical attributes and performance attributes. Physical attributes is simple like color or turbidity or appearance. Say, for example, a chemical is coming. Say one normal saline solution, more common constituent, a normal saline solution. When it is coming, As a quality person, the IPQA or in-process quality assurance team will look whether, okay, the color of the solution is correct or not. Then sometimes a colored solution comes, then he will check, okay, this colored solution is having a color index. So for example, 1.33 is the color index. So whether this color index is correct or not, they need to check it using a spectrophotometry. So that is one way. The another thing is the turbidity or appearance. Say if the SOP or the checklist says, okay, this particular solution should be clear. So we need to check whether the solution is clear, or is there any avidity, or is there any turbidity, or is there any flocculation, or is there any other some foreign particles are there or not. So then comes the third one, the appearance. Appearance in the sense, some chemicals are, liquid in nature. So some chemicals or some reagents which is used in the IVD test is in liquid format, whereas some is in lyophilized format or powder format. Even lyophilized powder format also two types. One is in a powder form, and another is in a cake form. Say, if my reagent needs to be in a powder form, I need to check, okay, the lyophilization is in powder form, which means my lyophilization was proper. Or if my reagent is in cake format or as a not powder format, lyophilized cake format. If it is a lyophilized cake format, which means that my lyophilization process is paka, or it is okay, it is within the acceptable limit. So these are the common physical, attributes we check while checking an IPQA or quality QC or QA in manufacturing process. And the next one is of very importance, performance. In performance, any reagent, it needs at least these 5-7 different performance characteristics or performance attributes. One is a method comparison, another is a sensitivity, second is linearity, then interference, stability, that is shelf life or shelf life. What is the shelf life and what is the open vial stability, or sometime they will tell us calibration stability also. So when my reagent, my IVD reagent, when it satisfies all of this without excluding any point, I say that, okay, my reagent quality is assured. So these are the major attributes. There are some other attributes also. We will come to that on a later stage. These are the attributes which is checked while controlling or assuring the quality of a manufactured reagent at a manufacturer site. Now let us understand these points one by one. Why we require a method comparison? The reason is that today if I am testing in lab X and I am testing my blood in lab Y, lab X is using a particular instrument, lab Y is using a different instrument. It doesn't mean that because of the change in the instrument in lab X and lab Y, my values should be deviate. No, my value should not deviate ideally or theoretically. Yes, as per the CLIA guidelines, there are some amount of variation permitted from when method to method is compared. It should be within this acceptable variation. If my reagent is not giving a value as compared to similar reagents available in the industry or similar methods available in the industry, which means that my reagent doesn't have a quality. So that is why method comparison. Even another one important factor is that now, somebody done a testing in say US, somebody done a testing in India, somebody done a testing in Sri Lanka. Person is same, only testing is different, only the environment is different. All these tests should be concordant. I will like to say it should be, the value should be concordant. Then only we can say that the testing is proper. I cannot say that, okay, you tested in US, there they are using something, then you get a result like this because we are doing in India. No. Whether it is performed in US, UK, wherever it is, whether it is in India, wherever it is, then, the value should be concordant. I'm not saying that, if US is 1, 2, 3, then here in India also it should be 1, 2, 3, or in Bangladesh also it should be 1, 2, 3. No, there are certain percentage of deviation is accepted by the CLSI or by the APLAC or ILAC company, international committees, then the value should be fall within this acceptable limit. So that is the meaning of method comparison. Now the other is sensitivity. Sensitivity now today, it is very, very, very important now because of the RDT. The sensitivity is very important for rapid diagnostic tests because sensitivity and specificity is very important because sensitivity means, what is my incidence of giving an improper or a false negative or false positive to a test. We will come to that on a later stage. Then the linearity. Linearity means, when I test a particular analyte using this reagent, till what level the reagent can detect without an error. Again, repeating the same thing. To what level a reagent can detect an analyte without any deviation. Now, the other is interference. Interference means, there may be some patients taking some medication. So, say for vitamin C, or the patient is having bilirubin. These are the commonly interferent substances in IVD testing. Then say something like then if they are having high fat content, for example, triglyceride or cholesterol level is high. In all those cases, this may interfere the assay based on the assay principle. So we need to assure that yes, with this reagent, there is no interference for a particular analyte or particular interference substance up to a particular level. Say, for example, if a patient is having bilirubin, if he is having jaundice, his bilirubin level will go high. When the bilirubin level will go high, there is always a chance of getting an interference in creatinine, which means the creatinine, whatever test they are doing, there is a chance of getting a small error. So we should check at what level the creatinine or the bilirubin will interfere to creatinine. Then comes the stability, shelf life. If I am telling that my reagent is stable up to 24 months, kept at standard temperature and pressure or STP, say it will work for 24 months. Then once it is open, then it will work for this many months. Once it is calibrated, it is work for this many months. So these are the claims we claim when we release any IVD reagents from the manufacturing facility. Now let us go one by one once again. How do we do this? So we are telling there are quality attributes. We are telling that there are certain checks is minimal. So how do we do this? So we have a QA QC, like it was already discussed during one of the earlier speakers. So quality, we have the quality assurance protocols and we have the quality control protocols. When these two protocols match each other, then we can assure that yes, the quality of the reagents is assured or falls within the acceptable criteria. So one is a proactive approach. Always quality assurance is a proactive approach, and the quality control is a reactive approach. One is finding fault or focusing on preventing, and another is finding. So one is preventing and another is finding the fault. Now let us see what are the methods used to prevent or find the fault in an IVD reagent. Before this, going further, we need to understand some phases in the analysis. Any analysis, whether it is IVD or any analysis which is going to take place, there are three phases. One is a post-analytical phase or post-analytical phase, then pre-analytical phase, then analytical phase, and a post-analytical phase. So we need to control, or we need to make sure that the quality is assured in all these phases. It is something like a link. It is something like a chain, I would like to say. The strength of the chain is the strength of the weakest link in the chain. Correct. So similarly, all these three phases are required, properly maintained, properly assured to make sure that yes, the final result is paka or correct. So each this point, we need assurance or we need checking points. Let us see what are the checking points and how do we these the checking point? How do we assure the quality? There are some flowcharts. I would like to say the 7 quality tools, flowcharts. There are check sheets, Pareto chart, a cause and effect diagram or a fishbone diagram, control charts, some histograms, then scatter diagram. These are the commonly used quality tools to make sure, yes, the quality of the reagents is within the limits. A flow chart, it gives what are the do's and don'ts on how to do the testing or how to perform the testing or what are the yes or no things to be taken while testing any analyte. A check sheet, actually, I would like to call it as a IPGA checklist. The check sheet is ensure that during the process, all the SOPs are, are taken care of. When the Pareto charts, cause effect diagram, these two comes in terms when there is a non-conformity or when there is an error. To find out, okay, if during the process somebody found that there is an error. So why there is an error? To do a kappa or do an RCA, we require this cause and effect diagram and the Pareto chart. Control charts, histogram and scatter plot, it actually comes into the real analysis part. While analyzing any analyte using any reagent, IVD reagent, we will understand, okay, my analyte, my value is this with when I validate my value with an existing value, or when I validate the value with a reference value, or when I validate my value with a traceable standard, the value should match or the value should fall within the limits. That is the usage of control charts. And a histogram, say for example, the common histogram what we use is a box and whisker B and W histogram, BW plot or box and whisker plot, which will show whether there is an outlier as compared to a reference matrix. Then the scatter diagram. The scatter diagram is a diagram which is used for the regression statistics to understand how my test or my analyte or my reagent is comparable to a reference reagent in real time. For that, we use a scatter diagram or we call it as a regression statistics. So please understand, when the IVD reagents are concerned, we use a lot of statistical process control tools to validate the reagent or to assure the quality. And there are many tools available. Commercial tools are available from NIST, from European Union. Many tools, softwares are available for doing all this kind of statistical calculations. Now, what are the guidelines we use? We are telling that okay, there are quality attributes. Okay, there are quality checks. Then there should be some guidelines. Correct. So there are certain guidelines which is used to do or which is used to analyze these quality attributes. For example, linearity. There is a guideline called as EP06A. For precision, there is a guideline called as EP05A3 for LOD or limit of detection. And there is a guideline called as EP17A2. Stability study, the famous Arrhenius equation. There is a guideline used which is called as EP25A. Method comparison. There is a guideline which is called as an EP09A2. These are all the guidelines given by CLSI or Clinical Laboratory Standard Institute from in US, which is given all these guidelines, and based on these guidelines, based on the statistics given in these guidelines, we assure that yes, my reagent or my instrument falls within the quality standards. Yeah. Now, to check the standards, there are some commonly used statistics are there. First one is the mean, that everybody knows that mean is, it's an arithmetic mean, number of variable, the sum of variables divided by number of variables gives you an arithmetic mean. Another is the standard deviation, which shows, how my value is dispersed. And the third one is CV percentage. This is very important. The CV percentage is very important when an analysis and IVD reagents are concerned, because even this gives more confidence to the laboratory pupil for using the reagents in their lab. The another is the zeta score or Z score, and third one is regression stat. Out of these 5 statistics, CV, Z score, and regression stat is very important for any laboratory testing. We will come to these statistics one by one. Mean is usually common to all. Now we use in SD or standard deviation. This is a formula for the standard deviation. So when we look at the standard deviation, one thing is evident, that when we do the analysis of the same sample, in a normal distribution, 68% of the values falls within plus or minus 2 standard deviation. Then 95% of the values fall within plus or minus 2 standard deviation, and 99.7% of the values fall within 3 standard deviation. This is the rule. How the rule comes is that when we do a testing, unless and otherwise it is a robotic testing, when a human or even an instrument do a testing, in a normal distribution, the distribution is normal. It's a normal distribution, and for a normal population, the curve will be like this, a bell type of curve. And the value 68% is nothing but the area under this graph. So 68% of the area falls under -1 to +1. That is why 68% is given, 95% is given, and 99.7% is given. These are the areas which falls under this graph. So this is taken as a standard. So usual cases, people, when we do a quality testing, plus or minus 1 standard deviation, if my value falls within plus or minus 1 standard deviation, my reagent performance is excellent. When my value falls within plus or minus 2 standard deviation, then there is a chance, it is a warning rule. I need to check, oh, what has happened? Something is wrong. So it's a warning rule. But we can accept that. But when my values falls within plus or minus 3 standard deviation, which means that yes, there is some error or something is not correct, we need to repeat the testing. So that is the importance of standard deviation in the quality testing. Then the other is coefficient of variation. Now I have given you 2 cases. So now it's an open questions to all of you. 2 cases. One is a glucose analyte, which is tested using a method called as a glucose hexokinase. And the second is an analyte, glucose, tested using a method called as glucose oxidase. These are the 2 different methods available in the industry. First one got a standard deviation is 4.8. Second one is got a standard deviation of 4. First one is having a mean of 120 and second is having a mean of 100. Now, the query is, which method is more precise? Can anybody tell which method is more precise? You can just type in the chat box. Yeah, both are same. Oh, great. Okay. Second one. Okay, very good. Both methods are the same. Glucose oxidase. Okay, please. Second. Okay. Oh, great. Yeah. Any more? Method B. Okay. Second, glucose oxidase. Very good. Very good. We'll just wait for some more answers. Then, we will come to the conclusions. Second, okay. Second, okay. Now let us understand this. Now, both are having a different mean, correct? One is having a mean of 120 XOBs, and the other is having a mean of 100. Whereas the standard deviation, the first method is having 4.8, and the second method is having 4.0. But please look at the mean. Both the means are not same. The means are different, correct? Now, how do you know which is precise then? For that checkup, as I told you, what is it actually for an IVD test? What we require is accuracy, precision, and resolution. So here, precision is very, very important. For accuracy is also very, very important. So how do you know which is precise? Now again, yeah, second. A lot of people are giving second. Now let us see, and I think 1 or 2% says both are same. Let us see this. Just a minute. If you look at this, to understand which one is precise, or we need to calculate the coefficient of variation or CV percentage. When we look at the CV percentage, both of this measurement is having a CV of 0.04%, correct? Both are having a CV of 0.04%, which means both are precise. So that is the reason the coefficient of variation is very, very important for analytical calculation, for precision especially. Or else, if you only look at the standard deviation, if you only look at the mean, there is always a chance of getting, misguidance. So here, CV, there is a golden rule. When the CV increases, precision decreases. When the CV decreases, precision increases. So that is a golden rule in CV. So people are looking for reagents or instruments which gives a lesser CV. If my reagent is giving a CV of 5%, whereas another competitor reagent is giving a CV of 7%, people will only go to this the reagent with a lesser CV. And for this CV percentage also, even as I told you, hardware is also very important. It is not a single listing of consumable. So CV is actually an amalgamation of both hardware as well as consumable, because hardware should be capable to detect even small deviations. Now the another is the zeta score, the Z score. The zeta score is calculated from the sigma or standard deviation, from the mean, or I would like to say the population mean and the population standard deviation, and also from the individual score. Why is zeta score is required? The zeta score is required. See, for example, I may be having a quality laboratory. Every manufacturer is having a quality laboratory, correct? So what is the importance of the quality laboratory? So they assure that the quality of the reagent or the quality of the instrument is correct, right? So how do I say that my testing is correct or not? Any answer? So for example, you are working in a quality laboratory. You are doing a lot of testing. How do you assure, yes, my testing is correct or not? Any answers? Oh, sorry.
We calculate the Z score, sir.
Yeah, by calculating the Z score, correct? By calculating the Z score, because the Z score gives you a excuse me, just the Z score gives you a confidence. Z score means it is a peer group. I would like to peer group, statistics. My score is X, and the population score mean is mu, and the population standard deviation is sigma. So I can assure that, okay, where am I standing in the peer group? So that is the importance of Z score. Even the Z score calculation can be performed within a laboratory. This Z-score is one of the best way to understand the differences of estimation within a lab itself. So for example, you are having lab A, lab B, and lab C. So you want to see whether all the labs are working properly. You need to use the Z-score. A Z-score which is less than 2, plus or minus 2, is highly acceptable, and a Z-score which is more than or equals to 3 is of problem. Now the other one is regression statistics. Here you can say we are comparing my reagent to another reagent. It's a comparative method. Comparative method is on the x-axis and test method is on the y-axis. Then we plot these two in a scatter plot, or we call it as a regression stat. It is a linear regression. If you look at the linear regression, you can see an equation and you can see an R squared, correct? Or this equation shows the relation between these two values. Then the R square shows whether it is a good fit or not. An R square which is more than 0.85 is good. So here they are correlating each other. 90 in simple statistics, 98% correlation is there in a different way. And if you look at the equation, even equation is also very simple, which means that my test is comparable to another test which is available in the market. So these are the regression statistics which is used for testing the quality of the reagents. Any questions so far?
Yes, sir. We have one question.
Yeah, please.
What is the reason for stating storage condition in the label of IVD in the board range from +2 degree to +40 degree? It is from Dr. Kashi Shankar Venugopal.
Yeah.
Okay.
Sir, the thing is that, these reagents, when you look at the IVD reagents, most of the IVD reagents comes into 3 categories. Category number 1, pure chemicals, like, the dichromate solutions or BCG solutions, like that. So there, the storage temperature, is maybe at room temperature is also okay, no problem. But when any reagent which is having an enzyme, it needs to be kept at maybe 2 to 8 degrees Celsius. Then there are some reagents or some RDT, like for example, now this rapid diagnostic test, say pregnancy test or some other test, or even some glucometer, something like that. It needs to be kept at even 38 degrees Celsius is also not an issue. But please understand, all these temperatures are based on the shelf study, Arrhenius equation. Say if I say that my reagent is having a shelf life of, say for example, 24 months at 2 to 8 degrees Celsius. So because it depends on the temperature depends on the characteristics of that particular reagent. We cannot keep a molecular diagnostic reagent in even at 2 to 8. It needs to be kept at -20, or else, because molecular diagnostic reagents contains pure enzymes, say BST polymerase or Tachman polymerase, all these polymerase enzymes. Because this polymerase enzyme is highly thermolabile, it needs to be kept at -20. Even any molecular diagnostics reagents, when it is transported, actually it is transported with dry ice and a data logger within it to make sure that this -20 transport is not lost. So the temperature at which any reagent needs to be stored is actually dependent on the reagent itself. It is not in a common. Okay, we can say that 2 to 45 or something, but each reagent is having its own, I would like to say, temperature or to keep, so that it will get the claimed shelf life. So that is based on the Arrhenius equation that we are coming on the part number 2. Yeah. Where do you begin if there is no reference or comparative method for a new biomarker analyte? Good question. It's a good question. See, for example, there is no, now, for example, this COVID, when the COVID came, we don't know about, there is no reference material available for the first 1 year because it is a new thing, correct? So if a reference material is not available for any analyte, we can do a comparison study between your analyte or your testing with another laboratory, another established or accredited laboratory testing the same test. So that is a method comparison. For example, you are doing the zinc. Example I'm giving, zinc. There is no comparative method or reference material is not available for zinc. Correct. So you need to validate whether your zinc is working or not. So what you do? You need to look for any other laboratories which is doing a zinc. If the zinc which is tested in X laboratory and when the zinc which is tested in your laboratory, you need to correlate using a regression statistics. It is available in the Excel. You check in the regression statistic if the R squared what we are getting or the line fit, the curve of the line fit, if the R squared is greater than 0.85 or if it is greater than 90, which means that yes, your analysis is perfectly all right. So that is the thing. Whereas if most of the cases, the reference method or the reference standards is available globally. There are two things. One is an NIST, National Institute of Standards and Technology, US, and another is an SRM, that is a standard reference material, that is also from US. Then another is ERM, which is European reference material. So if you can go to their sites, I think 99% of the analyte you can get reference standards, international reference standards, and this is a valid point. Actually, we are coming to this on the second part. It's a very valid point actually. Reference the comparative method, and the other factor is that see, if the comparative reference material is not available, then you can do a comparative reference method. There may be some comparative reference method. So there you will do, I would like to say, true positive or predictive values, positive predictive values and negative predictive values. So for example, if there is no comparative reference standard itself is available, so what do? You need to check it on a different method. Then if that method comes positive and your method comes positive, then it is matching. If there is no false negatives or no false positive, then it is matching. Like also we can validate. So it depends upon the analyte or it depends upon the method, whatever you are using. Please, any more questions?
One question.
Yeah, please.
Regarding this antigen testing, because in our places also, when one person is going for one laboratory for testing of this COVID by antigen, she was negative, and when she has gone to some other places and it was tested as positive. Great. So how it happens, sir?
Good question. See, molecular testing or especially the COVID testing, three different factors are there. Factor number one, with sample collection. See, the sample needs to be collected properly. If you look at the sample that collecting that applicator stick for nasopharyngeal stick, it is having a mark, correct? If any person, he is not, or while collecting the sample, if it is not inserting inside properly, then the sample or the viral load, I would like to say, in that particular bristles will be very less. And each rapid diagnostics card is having its own viral load. So, for example, I'm also having a RDT, I would like to say kit. My RDT kit, it is claiming, say, for example, 0.160 picogram. I'm just giving an example. So, if the viral load is 0.062 picogram, then only my kit will detect. Otherwise, it will not detect. Then the second thing is there any time gap? Say today you get negative. Okay. Maybe the viral load in your body is not that much. When you go to next day, maybe after even after 1 hour or even sometime even after 4 hours, if you go to the next laboratory, maybe the virus multiplied in your body like anything. So the viral load is improved or increased. So what will happen? When we do the sampling on the first time, you will get a negative. And when you do a sampling on the second time, you may get a positive, or vice versa also. It all depends upon the sample collection. You remember my previous slide, pre-analytical phase. This pre-analytical phase plays an important role, or sample collection plays an important role. I'll give you a typical example. So for example, I'm a diabetic patient. Okay, I went to a laboratory. Then he collected my sample in a common normal tube. We call it a cloth tube. Then they forget to do the testing for 3 hours. If I am diabetic, if I am having a high glucose value, after 3 hours, I will be normal. So they will report me as a normal person, even though I am a diabetic person. So here sampling is very, very important. And also you need to check what is the limit of detection. I think there is a slide on that. The limit of detection. Say for example, the RDT is having a LOD of 1 or 10, and another RDT is having a LOD of 5. Of course, the RDT with a lesser limit of detection will detect faster than the RDT with a higher LOD, like that. Yes, sir. Any more queries? It's critical to reporting and turnaround time. How do we calibrate maximum thawing cycle for 2 to ingredient? Yeah. This I will explain in the type of that shelf life. Now look at the again the quality attributes. Once again, the performance, that is method, comparison, sensitivity, and all. Let us understand the process flow, the manufacturing process flow. The manufacturing process flow is like this: raw material, in-process quality assurance, and finished good. There may be multiple sub-processes, but I have taken it all out. So raw materials, in-process quality assurance, and finished goods. These are the common manufacturing process flow in any IVD reagent manufacturing facility. So in each of these, we need to do assurance. So how do we do that? Raw material testing, for example, RM testing. So a raw material comes from the supplier, material supplier, or chemical supplier like Sigma or Merck or somebody else like that. So how do we share? How do you know that the RM supplied is correct? Because the person who is actually mixing this, he's not looking at the quality of the RM, because he's only looking at the RM and he's checking on the bottle whether it is approved or not. So how we do an RM or a raw material testing? Any raw material, first of all, we will look into the COA, or certificate of analysis. If the COA, all the attributes given in the COA is matching our requirement, then we feel that it falls within the limits. Then second thing is that there is an SOP. National, international SOPs are there for some certain raw material testing, and there are inbuilt SOPs based on our knowledge is there. So we will do a testing based on the inbuilt or based on the national or the guidelines, and we'll make sure that the RM is perfect. Once the RM is perfect, we will keep approved. We'll keep a sticker like approved sticker. We would like to say it's an approved sticker. So the approved sticker is placed. There are multiple ways. There are chemical ways. There are titrations. There are some enzyme trial batches. There are multiple ways of testing raw material based on the criticality of the raw material and based on the I would like to say reactivity of the raw materials. So the raw material is tested. Once the raw material is tested, then comes the IPQA, or the in-process quality assurance. Here, it is not only a fault finding. An IPQA is actually, I would like to say, it is a checklist. Say, for example, a particular reagent needs to be manufactured in a steel tank. I'm just giving an example, steel tank. So the steel tank needs to be cleaned properly. There may be some methods to clean the tank. So what we need to find out, we need to check whether the tank is clean or not. Yes, tank cleaning of the tank, tick. So how do you take a person or an in-process quality assurance team go there and they observe whether it is cleaned properly or whether the SOP is it is cleaned as per the SOP. So it is checked. Then the environment. It may be a class 10,000 or it may be a class 1 lakh. So whether it is in a class 10,000 environment or whether it is a class 1 lakh environment, they will check it once again. Similarly, the glassware used, whether it is a calibrated glassware or not. Here comes the calibration, whether it is a calibrated glassware is not. Then check, yes, calibrated glassware. So, for example, for measuring something, you remember the resolution kind of thing. Say, if you are measuring in kg, then we don't require a high grade, high sensitive, I would like to say, weighing scale. Whether when we are measuring the enzyme, we may require an ultra sensitive weighing scale. So whether the measurement is taken in the ultra sensitive weighing scale on a weighing table. So there are weighing table where all the gravitational force, everything is minimized. There is no shaking, there is no parallax. There is a particular hood under this. So all those things are there. Whether these enzymes is measured properly, so they will tick, yes, enzyme is measured properly. Whether the enzyme measured in an RH facility, yes, the enzyme is measured in an RH facility. So all those things are checked one by one, so that we can assure, yes, the process is proper. So that is these are the testing, or these are the checking performed at the level of IPQA. Then once the IPQA is checked, it comes to the final quality control and FGQC. Here they need to do a lot of studies or a lot of testing, because it is the last toll gate. I would like to say it is the last toll gate. There are 3 toll gates. RM testing is toll gate number 1, IPQA testing is toll gate number 2, and FG testing is toll gate number 3. Once this toll gate is opened, then the material goes to the client side. So here we have a lot of testing to make sure that the quality is assured. Now let us see what are all the testings required. Yeah. The question here is, first, as an FG quality team, we need to assure that, yes, my testing is correct. How do we assure that? We assure that using 2 processes. One is an internal quality assurance or IQA program. Another is an external quality assurance or an EQA program like Z-score, or participating in CAP accredited programs, or RCPA, or some other national or international quality assurance programs like that. We will come to that. The first one for any testing laboratory is IQC, or internal quality assurance. Correct? Now let us see what we do. Any quality lab should have an IQC chart. Any quality lab should have an IQC chart. Why an IQC chart is required? We need to make sure that, yes, there is no systematic error which is happening in our lab. There is no bias which is happening in our lab. There is no trend which is happening in our lab. There is no shift which is happening in our lab. Once we should make sure that there is not bias or there is no trend or shift which is happening within the laboratory, which means that, yes, the process we are maintaining in the laboratory is correct, or the process which is performed by the analyst or analysis is correct. It is in total. It is not for any reagent. It is not for any instrument. It is in total. It requires man, machine, and method. 3 M's. These 3 are there when we do an IQC chart. Now look at this. Any IQC chart, there will be a lower control limit and there will be upper compound limit. And there is a process average also. If you look at the point number 3, something happened out of control. This may be a special case. There will be something happened. Or if you look at the 4th onwards to 10th, there are zigzag variations, but all the zigzag variations are within the limit, which means that, yes, if you plot this graph in a histogram, you will get a histogram of a bell-shaped histogram, what we have shown in the standard deviation slide. So if the IQC or internal quality control is not proper, then whatever testing we are doing is not correct, because we ourselves is not confident on what we are testing. So this is one of the important facts when we assure quality of reagent. I am testing my reagent quality in my quality lab, or my analyst is testing this reagent. So how do the analyst or how do the team assure that, yes, whatever we are testing is correct? So that is IQC chart. So each analyte, there is an IQC chart. Each analyte. Then we will plot these analyte values in the IQC chart and we see whether it is within the limits. Then only we will test it, or else the entire testing is gone. So we do an IQC testing, and this is nothing but Shewhart control chart, or just there are some rules also, some Westgard rules and all that we will come to a later stage. So next we will see whether any error is there or not. This is an international standard Westgard rule, or I would like to say now 6 Sigma, Westgard, everything is putting together. The data we will check whether it is more than 3 standard deviation, whether it is more than 4 standard deviation, or 4 times 1 standard deviation, or 8 times same value. So there are certain criteria. We call it as a basic quality kind of QC criteria or Westgard rules. If my value is not violating any of these Westgard rules, which means that my quality is okay, my testing quality is okay, then I can release the report, release the reagent or whatever it is. So here the assurance is that my total error, or TEA is total allowable error, is within the limit. Now another is Iqc. As I told you, external quality assessment schemes. You look at this particular graph, it is alanine aminotransferase, that is AST, or it's a liver enzymes. You look at on the other side, your SD is 3.6. What is the best SD? 1.3 is the best standard deviation given. So when this particular enzyme is tested by n number of participants, so the standard deviation, or in precision, standard deviation got starts from 1.3 to 6.4. And you look at the percentage also. 90% of the participants falls under 6.4. 50% of the participants falls under 3.8. 20% of the participants falls under 2.9. And we, as a laboratory, we falls under from 20% to 50%, which means the quality of testing in my laboratory with that particular reagent is perfect. If anybody wants to know more, please ask me. Again, you look at the CV, coefficient of variation. Here also, the CV is from 1.2 to 6.6, and my CV is 3.8, which falls within 50%, which means my precision is good. But you look at the average bias. You look at the average bias. My bias is 10.4, but my bias is from 50% to 90% scale. So even though my standard deviation and coefficient of variation is good, it seems that I'm having a bias, or my value is biased. So this is the importance of Iqc. So first is an IQC, second is an Iqc of the same sample or same reagent. Then we get whether the quality of the reagent is proper or not. Any queries? Because all this testing is actually, based on statistics. So it may be sometime, it may be boring. I'm very sorry for that. Then what we claim, yeah, please.
By the you have seen anything like this? Any deviation in your reagent?
See, normally the deviation, once we test the reagent, we test that we have a standard deviation. Okay, if it is falling within the standard deviation, we approve. We have a coefficient of CV percentage. When it falls within the CV, we approve. And we have a bias. So usually this bias won't be there. Not a positive or negative bias won't be there. Even though the bias is there, the bias will be within this 50%, not more than that. Like that. So see, when we assure the quality of the reagents, based on statistics only we are assuring. These are the international statistics, available statistics. Based on this only we are assuring, yes, my reagent is perfect. Because I cannot just say that, after mixing everything, yes, my reagent is ready. But I need to do a lot of testing like this to make sure that the reagent falls within the, acceptable limits. So what we claim after quality assurance is that the method comparison, all those things. Now let us see what is a method comparison. Earlier we have seen one method comparison. Here, if you look at, you see on the left hand side a bias plot. A bias plot means it should not be, very constricted bias plot. It should be a minimal bias plot. You look at the bias. The bias is from minus 15 to 15. It is at a level of 300 units. So which is in acceptable range. And when we look at the other side, the comparative method to test method, the R squared is 0.99, which means my method is perfect or my reagent is perfect, as compared to reference method available in the industry or reference machine available in the industry. Yeah. Now, somebody was asking about the traceability, correct? So the traceability, this is the traceability matrix. There will be primary standards available in nationally or internationally. So from the primary standards, there are secondary standards or national standards of measurement. From the national standards, there will be primary standards or working standards and process standards. So it is a traceable matrix. So for example, if I say 1 kg, this 1 kg what I am getting needs to be calibrated to the 1 kg which is available in Kerala, which is calibrated to the 1 kg which is available in Delhi. The Delhi should be standardized to the 1 kg available in the Paris, and the Paris is the 1 kg is the topmost standard, correct? So that is the unbroken chain. That is called as a traceability matrix. And the same traceability matrix is available for IVD reagents also, like CRM or SRM. So we have an SRM material or CRM material with a definite value onto it. So based on that values, we actually validate all our reagents. Now, earlier, we were asking about the sensitivity and specificity, correct? So what is sensitivity is true negative, and what is specificity is true positive. And this is the, look at the other side. See, when we check the particular condition, head failure, 8 number came as positive, 2 came as negative. So out of 10, 8 is correct or positive. So here the sensitivity is 80%. Correct. When we look at the specificity, even not head failure also, 7 came as positive, whereas only 3 came at negative. So here the specificity is 3 by 10, that is 33%. So this is the importance when the such kind of rapid diagnostic test comes. So if you look at ICMR guideline for COVID-19, the rapid testing is more than 65% is the sensitivity or specificity is required. Another is linearity. I told you what is the linearity earlier, correct? So here linearity means, so for example, I'm doing a testing. When I'm doing a testing for 32 theoretical value, I need to get 32, which means it is linear up to 32. The same graph I have taken on the other side. It's a method for a method in milligram per deciliter. You look at after 35 or 32, the graph is coming down, which means after 32, there is an uncertainty. So when I validate my reagent, I am assured that, okay, my reagent is capable to detect up to 32 milligram per deciliter without any deviation. So that is my quality assurance. or that is my quality claim based on this. So for these kind of linearity studies, there are some controls available, or we call it as a linearity controls. So linearity controls are available globally, which gives an idea, okay, this, for example, C-reactive protein, the linearity control may be 320 or 32 milligram per deciliter. Example, this is what CRP. So when I look at CRP control with a linearity of 320 or 32 milligram per deciliter, what I will do, I will check the individual linearity metrics and I will see whether my reagent is detecting up to 32 perfectly. If my reagent is not detecting up to 32 perfectly, then I cannot claim my linearity is 32. So I need to claim a lesser linearity. There may be 30. If instead of 32, if it is not coming up to 32, I need to tell that okay up to 35. Oh sorry, up to 30. Or if it is not coming down, or if coming up, it is coming down to 20, I need to claim okay, the linearity is only up to 20. Now, the another thing is the stability. Yeah, here it is very important thing. Stability studies. The stability studies, what we do is based on the Arrhenius plot. So there is an ideal temperature. I think, everybody in this section is well versed with the stability studies and Arrhenius plot and all. But still, when we do a testing, our reagent, for example, earlier there was a query. Why do we require 228 or why do we require 245? Why there is a vast this thing? So I will take an example of say a liver enzyme, SGOT or glutamate oxaloacetate transaminase. SGOT and enzyme, I'm telling that this particular reagent is stable for 18 months. How do we claim that it is stable for 18 months? So I need to conduct a study. So I need to conduct a study where I need to keep these reagents in multiple temperatures. And there are certain guidelines also. In the guideline, it says if you are doing it at a temperature, let's say 2 to 8, you need to do an extended study or an accelerated stability studies. Because the particular reagent, I cannot keep it for a long time. No? There are real-time stability study and accelerated stability studies. So in our case, we need to do an accelerated stability studies. So if I'm claiming that my reagent is 24 months stable, I cannot keep 24 months for the same reagent to release. I need to take the reagent, I need to do some testing or accelerated stability testing and see how the testing is going on. And based on that Arrhenius plot, we come to a conclusion, yes, this particular reagent is stable for this many days or this many months or this many years. So in this particular case, the ideal temperature given is at 5 with a K norm of 0.001. It is a typical thing because ours is actually at 2. So I have just given it as 5 for some other purpose. So if you keep at 5, this particular reagent is stable up to 154 days. When we keep this temperature more, say instead of 5, we keep it at 10, then again the stability will come down. Correct? So similarly, for each parameter, each reagent, each formulations, we have a accelerated stability study reports, which make sure that yes, the formulation made is actually working or is stable at the claimed stability studies. Then comes the interference study. There are two different interferences are there. One is the bilirubin and another is intra fat or fat. As I told you earlier, bilirubin, when it is high, what will happen is that the reagent, the serum will have yellow color, and this yellow color in the serum, it will interfere with the creatinine using alkaline picrate method. There is one method available for creatinine, which is called as an alkaline picrate method, because the end product of alkaline picrate method and the bilirubin color is common. So what we need to do? We need to check the reagent, correct? So we will do a stability, we will do an interference testing study. There are interference substances available commercially from the international market. So for example, conjugated bilirubin materials are available. And we will do a recovery percentage. What is our recovery percentage? If the recovery percentage is not going less than say 90%, which means that, okay, it is not interfering with our reagent. If it goes less than 90%, which means that it interferes with our, I would like to say, our reagent. So based on the interference, we need to give a claim. Yeah, yes, this my reagent will not have any interference with bilirubin up to 30 milligram per deciliter concentration. So that is the interference studies. And this is a very crucial study. Now, today, if you look at, because of this particular interference, now many customers are now shifting to something called as an enzymatic bilirubin method. There's a different method is available in the market that is called as an enzymatic bilirubin, which is having more linearity as well as more sensitivity and very less interference. So, as a manufacturer, we need to make sure that our reagent is having minimal interference, or else it will create an issue. Second is an Intra Fat. Intra Fat is usually cholesterol, triglycerides. Any patient or any person having a higher cholesterol level or having a higher triglyceride level, their serum will be having a turbidity, highly turbid. So this turbidity can actually create issues while taking the reading. So what reading we are taking is that optical reading. So this turbidity can interference with the optical reading. Because of that, there is a chance of getting false elevated results or false, negative results also. So we need to make sure whether this is happening or not. And this intra-fat or intra-fat studies is very, very important when there is latex-based testing is happening. Because this fat, especially the triglyceride with more than 1,000, 1,200 milligram per deciliter, what will happen is that the particle will have a similar size that of latex particles. So there is always a chance of getting, I would like to say, high or elevated values when we do testing with the serum sample, the lipemic serum samples. So as a manufacturer, what we will do, every latex-based reagents when we manufacture, we will check whether this particular reagent is having an interference on this lipemia or not. One query came. Okay, please share the slide. It is not visible on the screen. Oh,
but it is visible for us.
It is visible on the screen. Okay, fine.
Yeah.
Yeah, okay. So this is an intra-fat. There is another one, ascorbic acid. This ascorbic acid or will interfere with many, I would like to say, analytes. We need to make sure, okay, ascorbic acid interference is not or not, there or not. Then some hormones, for example, thyroid, I believe it is biotin or something like that. Some particular drugs, it will also interfere. So we need to measure, okay, say ascorbic acid up to this much concentration, it will not interfere. If the concentration is more than, yes, it will interfere. So we need to study that also. So it is not that, the reagent needs to be tested for only the accuracy, precision, and resolution. No, it needs to be tested with these other quality attributes also, or else it will create issue at the client level or the customer level. And when we look at on the other side, excuse me, one more query it came. Name only displaying on the screen. Okay, sorry. Yeah.
No, it is not like that. No.
Yeah. Another is limit of detection. Sorry. Another is limit of detection. The limit of detection, as I told you earlier, there is something called as limit of blank and limit of detection. See, look at here. It is an LOD graph of, I think, one of our recent reagent only. There is a limit of detection for all, I would like to say, reagents. or formulation, I would like to say. And there is a limit of blank also. Limit of blank means, say if I am only giving, say, my albumin, only an albumin is giving, only reagent is giving. Sometimes there is a chance that it may give you an albumin value of 0.05 or 0.5. It is negligible as compared to the value which is what is called as a medical decision limit, or MDL. It is called as a medical decision limit. 0.5, it is not interfering. But if you earlier there was one query regarding why it is positive here, negative here. It all depends upon the limit of detection. What is the limit of detection? If the limit of detection is very high, there is always a chance of missing LOD. If you look at two lot reagents, here the 2-lot reagents is having a limit of detection 1.147 and a limit of detection 1.09, which is very, very, very concordant, which means that my formulation is correct. My previous formulation is correct. My today's formulation is correct. Now, when I go to the next formulation, these two, the today's formulation will be taken as a reference. So always I have a reference traceability of my reagent formulations. So we need to keep it. So this is also very important. We need to keep the reagents in our facility. So for example, I am claiming that my albumin reagent is stable for 24 months. 24 months plus 6. 24 plus 6. 30 months that reagent, one bunch of the reagents will be there with me as a retention reagent to check at any point of time till 24 months. And to do these kind of traceability studies. So to assure the quality today, we need to have a lot of traceability. Earlier it was not like that. Now why today it is more traceability is required, more this kind of what do you call it, analysis is required, because the rules are changing. The rules are changing means now if you look at LIC or Life Insurance Corporation or some insurance companies, they won't take any report which is from a common lab. Why? They need the report from an accredited lab, correct? An NABL accredited lab or an NABH accredited hospital, because they make sure that whatever report they are getting is correct. That confidence is there. So that is the reason they are going for an accredited lab. So what an accredited lab do? An accredited lab, as per the NABL 15189 or NABL 112, and as per ISO 15189 2012, there is something called as traceability. Traceability of the calibration, traceability for the reagent, then frequent calibration of the instrument. So all those things are required. Limit of detection, limit of blank. So as a manufacturer, I need to be ready to give all those documents, because documentation is very, very important today. I need to give all these documentations, all these documents to my client whenever they ask. So today, the assuring of quality of reagents as a manufacturer, for me, I need to do a lot of testing. Forget about the testing, multiple different levels of testing. I need to do a lot of, I would like to say, documentation. The documentation should be perfect. With the data, with the raw data, sometimes even when we are registering the reagents in some other countries or sometimes in the European Union and all, they will ask all the raw data. So we need to provide all this raw data, or else they will not accept. Previously, only the MSDS or certificate of analysis was only required. But today, we need to give all those, I would like to say, the DFM file with complete details. Then only they will accept. Because all these statistics, whatever we are discussing today, is responsible for the quality of that particular reagent. Without the statistical tools, we cannot say or we cannot assure, yes, my reagent is of good quality. So that is the reason of having this many statistics today. Now the new IVDR rule or European CE rule, everything is changing. So that because of the globalization. Now we feel that now more and more data may be required to assure the quality. We don't know. Because this is only for the instrument reagent. The instrument, you look at the instrument, again it is a different. It is much more points than the reagents. Yeah, some questions are there. Let me just go through that. Yes. For sensitive analysis, how about having lower limit of detection and upper limit of detection? Yeah, see, doctor, here the analytes with an LLOD on upper limit of detection, there is a guideline. I think it is given in my slide. There is a guideline for precision and trueness and limit of detection. So based on that guideline and with this analyte, for example, I would like to say hormones. Usually the hormones, the detection is in picograms because they're very minimal. So for a hormone, for a thyroid hormone, a TSH hormone, there is an international guideline. Okay, for TSH, the lower limit of detection should be this much, or the upper limit of detection should be this much. That is one international guideline. Second thing is that the manufacturer should claim that my lower limit of detection is this much and my upper limit of detection is this much, or my LOB or limit of blank is this much, based on the reagent formula. It is actually the inherent characteristics of the reagent. Again, the instrument used. If you are using, because certain instruments will be having more sensitivity because of the hardware. It is a hardware attribute. If I am using a highly sensitive reagent on a low sensitive instrument, I will not get the LOD. And vice versa. If I am using a highly sensitive instrument on a low sensitive reagent, I won't get the LLOD. So it is actually an amalgamation of both this. See, today, if I have given you an LOD of 1.147 and 1.09, I have taken it from my fully automated analyzer. This validation I have done on my fully automated analyzer, which that analyzer itself is validated using the IQC and using an Iqc program. So here complete traceability is there. Then only I can assure. I feel my answer is okay to you.
Okay, sir, one question, because in the regarding the drugs and pharmaceuticals, we have the references book like IP, BP, USP, and the extra pharmacopy and like that. So yeah, when it coming to the in vitro diagnostics, I think from IS or like that, any reference or the manufacturers methods are there, and some limit should be there internationally, like that.
Here the thing is that, you know, here there are certain guidelines by the NIST and the European Union and WHO. Some guidelines are there. So based on these guidelines only, actually we are doing this.
So like, you can because EP06 linearity, EP05 precision, like that only.
Yeah,
right?
like that. And there are certain reference material also. Like for example, SRM, certified standard reference material. This standard reference material will be having some, say, I'll just take one SRM. There was some SRM. Yes. That one SRM. It may be having maybe multiple, multiple analyte. Say, for example, an SRM. NIST reference material SRN 956B is traceable for calcium, cholesterol, then creatinine, then glucose, then urea, like that. So each of these analyte, there is a reference material is there. Either an SRM or ERM, and something other is there, is that SRM 186, like that. One number is there. One particular primary reference material is there. So Then sometime it is called as an IRMM, International Reference Material. So all this reference material will be having a value. So we will take this value as our reference. Then we calibrate our reagent or our formulation using this reference material. That is the process actually we are doing. And this reagent formulation also, there are some common formulations available, and based on the manufacturer, there may be some new iterations also. It depends upon manufacturer to manufacturer, like that. Then for some say controls, there are linearity controls or interference controls are available worldwide. We call this then some ICMS or LCMS. For example, creatinine. Creatinine, the standard is traceable to LCMS. So like that, certain controls are there. So we, as a manufacturer, we take the SRM or CRM and we actually calibrate, or I would like to say we instead of using the word calibrate, we make sure that our reagent is traceable to the international reference material. So that is a normal process which is performed at a manufacturer level.
Internally verifying your product with the international.
Internally verifying. See, internally verifying means when I verify internally, that my internal verification, for example, is actually traceable to international verification. How I will tell you. I am verifying all this thing in my laboratory, correct? My laboratory is verified by a third party, that is Iqc's party. It may be CAP, College of American Physician. It may be RCAP, Royal College of Australia. Then All India Institute of Medical Science, CMC Vellore. Then there are some, then Biorad. There are a third party, these accreditations of third party Iqc's are available. When I participate in this by Iqc's, when my value falls within the standard deviations or CV percentage or bias as per the international, which means that my laboratory setup or my quality setup, not laboratory setup, my analytical setup or my analysis is within the standard, limits. So that way we
like the proficiency testing.
Pardon?
Like the proficiency testing.
It's a proficiency testing only. It is a proficiency testing. Otherwise, if you're not doing a proficiency testing, then whatever you said is correct. We don't have any control on that. Mostly it is proficiency testing. There are certain standards available, like it is available in the WHO and say for example, now when we look at the COVID, all those COVID sequencing, genome sequencing is there published by the WHO. Once the WHO published the genome sequencing, anybody can do that sequencing. Even we also did the similarly. We also taken that sequencing only. It is a public document. WHO published document, which is a reference standard for us. Like that.
Another question is coming. Which are the major forces involved in biomolecular interaction between antibody and antibody, and how is it evaluated? It is from Girish Vagela.
Which are major forces involved in biomolecular interaction between antibody and how it is evaluated? See, antigen-antibody reaction. See, when we look at any reagents, I will only talk in terms of reagents. Antigen-antibody reaction, there are three different ways. Because antigen and antibody itself, we cannot see with the naked eye. There is no color change. Nothing is there. So what we do? We do either in a competitive assay or a sandwiches. Correct. So, if it is a pure antigen-antibody reaction, any antigen which combines with an antibody, an agglutination will be formed. Then the rate of agglutination or turbidity form is directly proportional to the, I would like to say, to the analyte. Say if an antigen is analyzing, then antigen excess, or antibody is analyzing, then antibody. That is type number 1. Type number 2, visible antigen-antibody reaction, like latex, latex test. When we do a latex test, even visible antigen-antibody reaction, I would like to say it is that blood grouping type of testing. So we can visibly see that. There, actually a flocculation is forming. The third one is latex enhanced immunoturbidometer. So to detect the latex form, the antigen antibody reaction formation, we give a third party or a latex. The latex which is coated with antigen or antibody, or it may be a sandwich type of antibody, or it is a competitive type of antibody, so that the antigen as an analyte or antibody as an analyte can be detectable. So these are the 3 or 4 methods which is used for detecting an antigen and antibody reaction. Whereas the antigen and antibody reaction itself is dependent on multiple factors. Factor number 1, whether it is a polyclonal or a monoclonal antibody cocktail is, or antibody cocktail is used in the reagent. That is one thing. Because monoclonal will be more specific, polyclonal sometimes it won't be that much specific. That is number 1. Then there is something called as an FC specific antibody. So whether are we using an FC specific antibody, then it is something different. Then whether there is an antigen excess or antibody excess is there, then the reaction will not happen properly, because it is called as, it is called as Prozone Effect or Hooke's effect. It is already there in the textbooks. Prozone Effect or Hooke's effect. The antigen antibody, one antigen will capture one antibody or two antibody, or one antigen to one antibody, or antigen clumping will be formed. In a reaction, if the antigen is excess, reaction will not happen properly. In a reaction where the antibody is excess, again the reaction will not happen properly, because the number of interactions will be minimal, or the antigen itself will prevent other antigens to interact with the antibody, or the antibody itself will prevent other antibodies to interact with the antigen. So if the antigen excess or antibody excess is there, then also the reaction will not be happening properly, and which is called as a Prozone effect or Hooke's effect. Actually, when we look, go back to this slide, yeah, here the linearity. When we look at the linearity in an antigen antibody reaction is actually because of the Hooke's effect or Prozone effect. Because when you look at here, after 32, it is curving down, means the reaction is not happening as linear, because the antigen is excess or antibody is excess. So that is why the reaction is coming down. Yeah. What is the other question?
Sorry, it is
RIA or ELISA. RIA or ELISA. RIA or radioimmunoassay is having its own merits and demerits. ELISA or enzyme linked immunosorbent assay is also having its merits and demerits. It depends on the analyte you want to detect. If the analyte you want to detect, if you are looking for highly sensitive analyte, then obviously you should go for an RIA, or you should go for a CLIA or IFA. There are 3 things: RIA or radioimmunoassay. The upper level is CLIA or chemiluminescence immunoassay. Then the next is IFA or immunofluorescence assay, and the fourth one is eCLIA or electrochemiluminescence immunoassay. These are highly sensitive assays. Okay, because it depends upon the analyte you are looking for. Whereas in ELISA, it is less sensitive as compared to the RIAs or other immunoassays. Because ELISA, you are actually looking for the color. It is a color formulation. When you add this topping solution, you can even see the blue color or yellow color, green color, depending on the analyte. Correct. And the color intensity you are checking. Whereas in an RIA, it is the flow or glow. There are 2 types of assays in CLIA. One is called as the flash and another is glow. When we add the final reaction reducer, a glow will be there, like chemiluminescence. A luminescence will be there, and the luminescence is directly proportional to the amount, and that is in glow based CLIA, flash based CLIA. In a glow based CLIA, when we add the reducing agent, it will start glowing, and for some time being, it will reach the maximum glow, and the maximum glow is directly proportional to the, or the rate at which the glow is happening is directly proportional to the analyte. So that is also more sensitivity. Similarly, immunofluorescence. All those things are for highly sensitive assays. If you are looking for a highly sensitive assay, definitely you should go for an RIA. Provided you have a method, you have a kit available for RIA for that particular analyte, or you should look for some other methods which is available for that. Yeah. Yeah. The other question is that.
Free license also for RIA.
Yes. RIA, I believe that you still required from license from BARC. But the thing is that, madam, now RIA is overtaken by ELISA. Sorry, not ELISA, CLIA. Now most of the analysis, if you look at RIA maybe in the late last 10-15 years ago, the RIA is coming down. Now only used in research. Yeah. Doctor is telling it correctly. Only this RIA is used in research, not in the common practice because of its demerits, because it is having radiations and all. So because of that, it is not used. So that is the reason we require as license from the BARC. Yeah. So this is the limit of detection, and now we discussed about multiple quality attributes. Correct. We discussed about method comparisons. We discussed about limit of detection. We discussed about the reference ranges. We discussed about the method comparison, this CV percentage. We discussed about a lot of things. Even the temperature at which a reagent or a formulation needs to be stored. We discussed all those things. Now my question is, how the customer know all those things? What actually the customer is getting? Customer is getting an information sheet, correct? So in those information sheet, are we giving all those things? Or what is the importance of giving all those things to the customer? Now let us go through one of the sheet for information. See, this is one instruction. I think everybody can see this. Let me check. It is visible?
No.
Now look at this instruction for use, which is for a liver enzyme, alkaline phosphatase. Intended use. You look at here, linear. So here linearity is mentioned. I will just
Not seen.
Yellow. So here linear up to 2000 U bar liter or international units per liter. And you look at here traceability. We talked about traceability, kinetic method traceable by recommended by IFCC, which means that this method is traceable to IFCC method. This method is having a linearity to 2000. Correct. Now, when we come back on the next stage, you can see that. See, once
Not seen any slides.
We are not seeing, sir. We are not seeing.
You're not seeing. Okay, then okay. Then I will stop share, and I need to share. Once again, I need to share. Just a minute. Yeah. Where is it? Yeah. Yeah. Is it visible now?
Yeah.
Now it is.
Okay. Now you look at this particular IFU, instruction for use. In this IFU, whatever we discussed is given, because that is the current trend. Whatever we have discussed in our presentation needs to be given to the client. Now look at here. Here the traceability, that is kinetic method recommended by IFCC. It is linear up to 2000. So linearity is mentioned here. When we look down, you can see the sealed reagent is stable up to so and so, protected from light. Then see, once open, the reagent is stable up to 4 weeks if contamination is available. This is the stability based on our equation. This thing is the calibration stability is for 7 days. And just a minute, reagent deterioration. Now you look at the interferences. No interference for this reagents. Bilirubin up to 10 milligram per deciliter. Hemoglobin up to 1000, ascorbic acid up to 50 milligram per deciliter. So interference study. Now, when we coming down, if you look at the quality control, you look at the quality control, you look here, one is the linearity which is given, that is a performance. Another comparison study, you see, a comparison study has been performed between agape reagent and other international available reagents, yielded a correlation matrix of 0.99, which means that it is 99% it is correlating. So which means that this particular formulation, when I release from my manufacturing site, is meeting all the quality standards as per the international thing. You look at the precision here, you look at the CV here, 0.89, 0.55. There are two levels. That one is an intra-run and another is an intra-run. It is between run and within run, I would like to say. And if you look at the other one, lower detection limit is 6 U-barrel, which means that my reagent is capable of detecting alkaline phosphatase as low as 6 units per liter in the blood. So whatever we have discussed, all those points are given in the IFU, which is a new standard. If I am going to send these reagents to any countries or any customer, any quality conscious customer, without these attributes, quality attributes, they will not accept. So as a quality person, we need to do all this testing within our quality lab to make sure that yes, my reagents is meeting national or international standards. Yeah, so this is the last slide I would like to say. Now, further questions from anybody, please ask.
You are there, Dilip?
Yes, sir. No questions in the chat box. So, the sample tested from patient with different disease condition. So, how the interpretation of result with the interfering factors?
Can you repeat?
Sample tested from patient with a different disease condition.
Huh.
So, how the interpretation of result with the interfering factors?
See, usually the most of the interference substances are taken care by the manufacturer, reagent manufacturer. Say most common interfering substances is ascorbic acid, then bilirubin and intra fat and hemoglobin. These are the most common interfering substances in the blood. So these 4 things are taken care, and there won't be a drastic change in the analysis unless and otherwise the reagent is very bad. There won't be drastic change. Yeah, I think the other question. Yeah.
So
I think
2 hours of presentation. Yeah.
Yeah. That, I'm glad that within the 2 hours we have stopped it. Yeah.
Yeah, because there was more persons from the drug analytical lab, government analytical lab, and from the chemical examination lab, and more professors and the students from Al Shifa College and Sri Krishna College, then and the Cwntral Medical College, and many faculty members from all of the places. Because it's a different subject for us, because we are concerned with the drugs and pharmaceuticals and everything. So, like enable accreditation, so your concern, your organization is also enable accredited?
No, enable actually do accreditation for testing and calibration laboratories, where the laboratory needs to release result to patients. So here we are not releasing, as a manufacturer, we are not releasing any testing result to the patients. So at present scenario, Even our laboratory is not a calibration laboratory. Ours is a internal testing lab. So that NABL or is not, or QCI accreditation is so far not required. Maybe if the new rule comes, if somebody says, you need to be NABL accredited, then that is a different thing. So that is the reason we are participating in more IQOS programs or proficiency testing programs as a manufacturer. But as a manufacturer, we are participating in many proficiency testing programs from India and abroad in multiple segments like hematology, then clinical pathology, then specific protein, diabetes. See, please, I'm proud to say that our reagents are certified by NGSP, National Glycosylation Standardization Program, which is a European body, which is actually a European body which gives accreditation for the HbA1c for diabetic control. And our reagents and instruments are this NGSP.
Share, Sanjay.
It's a wonderful presentation. Sir?
Can share the presentation.
We can connect one-to-one to discuss about your experience on that ROC curve, na?
Yes.
Yeah, ROC curve, it is a different thing. Yeah, okay, ROC curve, because actually these are the stats. So, Doctor, what we can do is that there is some statistical SPC tool available. One is called as Analyze It, and another I forgot the name. Statis Pro. Yeah, Statis Pro and Analyze It are the two different things available. So we can make use of that, because ROC itself is a little deep subject. We need to understand, because it depends upon whether you are doing a population or we are going to do a stat for a sample or a population, because it depends upon all those things. Yeah.
Can share the presentation, Sanjay.
Yeah.
So next time we can come with the instruments, different types of instruments used for the testing of IVD reagents.
Yes. Because instrument, again, I'm telling you, instrument also, the stat is very, very, very high. Statistics is very high. And there it is connected with, I would like to say, IEC, that is International Electrotechnical Committee. IEC standard, one standard is there, IEC 610101 and IEC 610102. These are the two standards, and there are some other standards is also available there. So we need to discuss that in detail. We will discuss that, no issues.
We'll make it after 2 months' time, Sanjay.
Yeah. We need to and make it in 2 months' time. Yeah.
Saji, any comments, Saji?
Yes, Dr. Satish, I can say it is an excellent presentation, and all biostatics and everything he has included. And see, particularly all the laboratories working in our country involved in the testing of the drugs or medical devices. They should learn what he has spoken. This is excellent. I was just going through each and every slide what he has shown. Particularly that biostatical data, that is very important. It is not a simple job. And now see, it is in time of artificial intelligence also. That AI is playing a very important role in pharmaceutical industry, in drug development, in drug discovery, in manufacturing, and so on. My special thanks to Mr. Sanjay sir. Sanjay, this was an excellent presentation. Really, I can see I can talk to Dr. Samani also. At least in a group of drug controller, we can invite such a speaker.
Definitely, sir. Definitely.
Yeah, thank you very much. Thank you very much.
Thank you, sir.
Yeah.
Now, or to Dilip, any comments? Dilip, anything to convey?
So, no, sir, because almost all persons have conveyed their feedback that it is an excellent presentation. They never come across like in a webinar other than the analysis of drugs and pharmaceuticals. This is a new subject to them. So hearty congratulations for Sanjay sir for giving a good presentation.
You'll share the link afterwards
We'll be hoping
for
you
repeating it. Yeah,
to come
share
with
the link.
it.
For Sonu?
Okay, sir. Over to Sonu.
Thank you, sir. That was
One remark.
Yes.
Dr. Satish, I can say, I don't know how the Kerala people are very intelligent, either to do coconut water or something else. I don't know. But I have been in our chief secretary, Mr. Kuti, in Delhi.
Yeah. Yeah.
He was very intelligent officer, what I have seen in my life. So hats off to all the participants from Kerala states. Thank you very much.
So we were
What he
waiting
says is-
to see you. Sir, we were waiting to see you in Kerala.
Yeah, I was supposed to come, but I changed my program. I went to, Northeast this time because due to some COVID issues. But
Next time
definitely,
you can come. Always next time.
Definitely, I will plan to come to Kerala.
I told
I
sir,
will call my wife also. Madhu, ek minute aa gayi. My wife is there. She is very much willing to come to Kerala.
Yeah, yeah.
Sir, before the next
so I told him while in Delhi, sir.
Yeah, hello. Hello, madam.
before the next wave, you please plan, sir.
Drop in anytime. Welcome.
Sure,
Welcome,
sure.
madam. Anytime to
Definitely.
Kerala. Well, take care. Please. Okay.
Thank you very much for invitation.
Thank you. Yeah, Sonu.
the feedback of all the participants in the chat box itself is showing that it was a splendid virtual learning experience. So on behalf of all the participants, I'm taking this opportunity to thank the very knowledgeable speaker of the day, Sanjay sir, and Dilip sir for the effective moderation. Now we are at the official end of the program, and I'm so happy to invite the most dynamic General Secretary of Kerala Pharmacy Graduates Association, Mr. P.U. Abdul Nasir sir, Production in Charge, Kriti Pharmaceuticals Limited, Chalakudi, to propose the vote of thanks. Over to you, sir.
Thank you, Sonu. Very good evening to all of you. Respected chairman of today's program, Dr. Satish Kumar, Dr. Adil Kumar Nasa, Mr. Jain, Dr. Sri Kumar, Mr. Sanjaymon, Mr. Dilip, Mr. Matthew Kokkat, and all other dignitaries, participants. It gives me immense pleasure and honor to propose vote of thanks for today's program. Actually, it was a very brainstorming session by Mr. Sanjaymon. First of all, let me propose vote of thanks to Dr. Satish Kumar sir. He is the eminent and dynamic president of IPGA Kerala branch, and he's a great visionary, and he is our guide in all our organizational activities. I propose my sincere thanks and gratitude to Satish sir.
Thank you. Thank you.
This is our today's inaugurator of the program and our chief guest, Dr. Adil Kumar Nasa sir. As we know, he is very close to us, and he is a great visionary. He is leading the entire pharmacy profession of the country, I can say. Sir, a sincere and warm gratitude and thanks to you for your presence in the
Very
program.
much. Thank you very much. Thank you very much.
Next is today's guest of honor, Mr. Jain PM, the Honorable Drug Controller of Kerala. As we know, he was the immediate General Secretary of Indian Pharmacy Graduates Association and a well-wisher and supporter of the entire profession. I propose our vote of thanks and gratitude to Jain sir. Next is Dr. P.K. Srikumar sir. He is the eminent president of Kerala Pharmacy Graduates Association, and he is the guidance and he is holding the leadership in the pharmacy profession in Kerala. I propose vote of thanks to Dr. Srikumar sir. Next is Mr. Sanjaymon. My goodness, it was an excellent, fantastic class by Mr. Sanjaymon. His clarity of the speech, his body language, and what all I should say. So Thank you very much, Mr. Sanjaymon, for an excellent class, and I extend my sincere thanks and gratitude to you. Next is, I can say, Mr. Matthew Kokkat, sir, who welcome today's gathering. Matthew, sir, is a good professional, and also he was holding the key position in our organizational activities, and also he is the MD of Kokkat Pharmaceuticals. I propose vote of thanks to Mr. Matthew Kokkat, sir. Next is Mr. Dilip, who is the moderator of today's program. As we very well know that he is actively involved in all the organizational activities of the pharmacy professional bodies in Kerala. And also he is a well-wisher, and I propose my vote of thanks and gratitude to Mr. Dilip ji. Next is our host of the day. Sir has come immediately when we called him. I extend my warm wishes and propose vote of thanks and gratitude to sir. Next is the today's anchor of the day, Ms. Sonu Benny. I propose vote of thanks to Sonu also. We can see many of the analysts in the drug testing lab here. I especially mention all of you, and thanks to all of you for your kind presentation, especially Mrs. Sudha, head of the drug testing laboratories, Mrs. Pushpakumari, Ernakulam lab head. I propose vote of thanks to all of you, and also I propose vote of thanks and sincere gratitude to all the participants who all gathered here and make today's program a grand success. Finally, I request all of you to be very active, and requesting all your participation in all the forthcoming programs of IPGA and KPGA. Thank you. Namaskar. Good night.
Thank you so much, sir.
Yeah.
And thanks to Mr. Abdul.
Thank you, sir.
That's very important.
Okay.
Yeah. Nasaji, every last Sunday we are planning to have one program for IPGA Kerala hereafter. Every last Sunday. We'll be
You fix it. See, you fix a date. I will be there in all your programs.
Thank you so much. Yeah.
Thank you, sir. Thank you.
Okay. So great day. Thank you all of you. I think it's time for us to.
Good night. Good night.
Okay. Thank you all. Good night, everybody. Good night. Good night, everybody.
National anthem, sir.
National anthem.
Yeah.
Thanking you all once again on behalf of IPGA, Kerala State Branch, IPGA Central and KPGA. Let's raise for national anthem.
जनगण मन अधिनायक जय हे भारत भाग्य विधाता पंजाब सिंध गुजरात मराठा द्राविड उत्कल बंगा विंध्य हिमाचल यमुना गंगा उच्चल जलधि तरंगा तव शुभ नामे जागे तव शुभ आशीष मांगे गाहे तव जय गाथा जन गण मंगलदायक जय हे भारत भाग्य विधाता जय हे जय हे जय हे जय 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 हे
Have a great and safe time ahead. Thank you so much.
Okay, thank you, Jayapalan sir, for supports. Jayapalan sir, all supports you have. Thank you so much.
Thank you, sir. Thank you. Shall I end up the session?
Yes.
Thank you, sir. Thank you,
Thank you
Jayavarman,
all.
sir.
Thank you all.
He's
Good
my
night.
teacher. He's my teacher.
Thank you.